Good morning, everyone, and welcome to our webinar. This is the second webinar in the series of 3, and it's on process and recent outcomes related to digital sequence information on genetic resources under the CBD. So my name is Charlotte Germain Aubry. I am in the Access and Benefit Sharing team at the CBD, and welcome. Just a note, I'm going to do a little bit of housekeeping and then give the floor to David Cooper. Just a note, this webinar is being recorded and will be posted online. And it will be made available on our website in the next few days. This webinar is in English only. There are no translations into other languages. There will be some polling and survey. There will be 2 questions at the beginning, and then when you log off, there will be 4 questions actually. And so please take the time to log off a couple of minutes early before your next meeting or your next task to be able to answer those questions. They're actually very important to us and will be very helpful in guiding the 3rd webinar. There is only a chat and a questions section. There is no chat section in GoToWebinar. It's a little bit different than what you might be used to. Because of the number of participants, all microphones will be muted, so no participant can come in and ask a question orally. So please put your questions in the question section. However, the questions that you post will only appear to everyone else once they are answered. Since there's a lot of people on the webinar today, I will ask you to please bear with us. We have people who will read your questions and direct them to the panelists who are most appropriate to answer them. And once the panelists have a chance to answer them, they will appear to everybody. So it's going to take a little bit. Please be patient, and we might not be able to answer all questions just because of the number of people on the webinar today. If you write little words like, hello from Peru or something like this, we will not be able to see it. So we're all very happy to see you, and we welcome you to our webinar. And we hope we're going to have a fruitful discussion in this webinar. Now, I will give the floor to David Cooper, who's Deputy to the Executive Secretary at the Convention for Biological Diversity. David, would you like to?
Thank you very much, Charlotte, and welcome everyone. Good day. So I'd like to welcome you on behalf of Elizabeth Maremma, the Executive Secretary to the CBD, and to the whole Secretariat team, the ABS team here, to this second in the series of DSI webinars looking at digital sequence information. We know that this is a critical issue in the convention, in the protocols, the Nagoya Protocol, the Cartagena Protocol, and actually it's relevant to many other agreements and processes in agriculture, in health, in the oceans negotiations. We know also it's going to be a critical issue in the negotiations that are ongoing to develop a post-2020 global biodiversity framework. And so we thought it really important that we have a series of webinars to make sure that all the information is shared and people can participate in these discussions and consultations effectively. So as many of you will know, because you participated in the first in the series of these webinars, which took place last week, we had a webinar last week organized and hosted by the ABS Capacity Development Initiative, and I'd like to express my appreciation to them for working with us on this introductory webinar, which provided a lot of background technical information on DSI, on the production, distribution, and use of DSI. In this webinar today, over the next couple of hours, we'll be focusing with you on the results so far of the process being carried out under the Convention on DSI, and in particular on the outcome of the ad hoc technical expert group that was mandated by the COP in decision 1420. And the ad hoc technical expert group actually took place in March this year, and we'll hear more about the results of that in a while. And then just to note that there will be a third webinar. This will be organized in the new year, and we'll have there the participation of the co-chairs of the post-2020 Global Biodiversity Framework. And the purpose of that webinar will be to share very informally some of the policy options that are emerging to address this issue of DSI and how it may link to the broader issues, including the post-2020 framework. So stay tuned for more information on that webinar when it becomes available. So today, as I mentioned, we'll be looking at the work of the ad hoc technical expert group and the process that was mandated by the COP. As you know, the AHTEG was in fact mandated by the COP to develop options, especially for the operational terms of how we describe DSI. What does it mean? And what would be the implications of different ways of grouping DSI? And this would be designed to provide more conceptual clarity on the issue, and then look at also capacity building needs that are related to DSI. The AHTEG, I think, made some very important strides in developing options for how DSI can be understood, and put forward a number of groupings of different categories of information that may be considered DSI in different circumstances. And we hope very much that this will provide conceptual clarity and assist the further discussions and the negotiations. I would like to thank everyone involved in that AHTEG, all the members of the AHTEG, and in particular the co-chairs, Chris and Leticia, who are with us today, and we'll be talking in a while, as well as all the expert presenters who I'm very pleased are also with us today, and will be guiding us through some of the issues that were discussed and addressed at that AHTEG. So I think without any doubt, the work of this AHTEG has generated some good options. You will see that perhaps the relevance of those different groupings of how we look at DSI might vary depending on the issue being addressed, and when we come to benefit sharing, the approach that may be taken to benefit sharing. It is still clear, though, that there's still more work to be done before we will get a complete common understanding, a complete consensus among different countries, among different parties, among different sectors on how to handle DSI issue and how it relates to the three objectives of the convention, and in particular the objective on benefit sharing. How can we make sure that those objectives are upheld, that we have progress to those objectives, that we have fair and equitable benefits from the use of genetic resources, while also making sure that we don't impede access to data, we don't impede scientific and technical progress, that scientists and researchers still have access to the work, to the data they need. And that this is why we need to make sure we involve all the actors, all the stakeholders in this process. It will be an iterative process, and we hope that the results of the AHTEG will be useful, if you like, in this sort of tick-tock between the science side, the more technical side, and then the more political side of these discussions. The results of the AHTEG are meant to feed in to the third meeting of the working group, the open-ended working group on the post-2020 framework, the working group that was established specifically to negotiate the post-2020 agreement. Given the ongoing pandemic and the restrictions on organizing meetings, given the fact that we need to hold meetings of our implementation and science bodies, SBSTA and SBI, before that group can meet, it's still some time before that meeting will take place. Of course, I think everyone is a little bit worried that and concerned that we still make sure that we maintain progress, maintain momentum on the discussions. And again, hence these webinars that can perhaps help to do that. So I hope that you'll find today's webinar informative and useful. I look forward to the presentations myself, and we look forward to your questions in the Q&A sessions. Again, I'd just like to thank the ABS team in the CBD Secretariat and the co-chairs and presenters who you're going to hear from today. Thank you very much. Back to you, Charlotte.
Thank you very much, David. That was a very nice, warm welcome and putting things into context of, the policy, the reality of the policy process, and also the series of the 3 webinars. So thank you very much for that, David. I just wanted to say a little note about the questions that you post in your questions box. The panelists will answer questions that are relevant to this webinar. So if you are asking questions about technical overview, for example, this was covered in webinar 1, which is now posted online on YouTube, and we can maybe put the link in the chat for everybody to have it. And then the webinar 3 will be concerned with policy options for DSI and access and benefit sharing. So if you have questions about that, maybe hold them until the 3rd webinar. This is when we'll be talking about this. We're getting a lot of questions, so we're trying to make sure that all the relevant questions to this webinar get channeled to the panelists and answered. So before we get started with the webinar, I would like to launch a very quick poll that should appear. And it basically is asking you where you are situated, located, or which kind of region of the world you are representing. So I'm just going to leave this for a few seconds so that people have a chance to answer, and then we can actually look at it together. So I'll give you a few seconds. Okay, I think most people have had a chance to answer. And okay, so you can now see the poll results. So we have a lot of people from Western Europe and others, which includes North America and Japan. We have a significant number of people from Asia, which I'm very thankful for because I know it's really late for you, the Asia Pacific region. So welcome and thank you for being here. We have a good representation from Latin America, the Caribbean, and also Africa. and then also some people from Eastern and Central Europe. So we actually have a fairly good representation of the globe, and I'm very pleased that we can say we have such a broad representation. Then I'll give you a second question, also fairly quick. And it's basically about, who you're representing when you're attending the webinar today in your function, in your role. Who do you identify with? So you're going to have government, intergovernmental organizations, academia, collections, museums, the private sector, or civil society organization, NGO, indigenous peoples organization, or others, if you cannot recognize yourself in any of those classifications. So I'll leave this up a few seconds and then we'll look at it together. Okay, so it's been 1 minute, so I think we have most people, and now you can see the result. So we have a very large representation of government and intergovernmental organizations. So a lot probably of our parties are represented. So welcome and thank you for being here. We have a good representations from researchers, scientists, academics, people working with collections and with museums. So welcome as well. And then we have an equal representation of civil society organizations, NGOs and indigenous people organizations, and the private sector. And then a few of you cannot identify yourselves with these categories. So I think we'll also have, a good representation, a little bit of everyone is here, and that's very good to see. I hope it's going to really launch into interesting discussions and questions for our panelists and our co-chairs. So thank you very much for answering those questions. Now, I'll give you a very brief overview. So you've heard from David Cooper, and we're about to hear from Christina Tabulchanas, who is a team member in the Access and Benefit Sharing at the Secretariat for the Convention of Biological Diversity. And then we'll go into the key findings of the peer-reviewed studies. So each of the studies will be presented by one of their authors. And then we'll have a Q&A session on those studies for our panelists. Then the second part of the webinar will go into the AHTEG report and the key outcomes from that work. And the co-chairs will be summarizing this for you. And we'll have a Q&A session after that to discuss with the co-chairs on this particular report. And then we'll have a wrap up from the head of the ABS unit at the Secretariat. So I will now give the floor to Christina, a team member at the ABS team in the Secretariat. Christina, are you ready?
Good morning, everyone. I'm trying to share my screen here. Are you able to see it?
Yes, I believe so, yes.
Okay, perfect.
And if you can put your camera on.
My camera on. Okay, here we go. Great. So good morning, everybody, and good afternoon and evening for many of you. Before we get into the good stuff, I would like to share some background information about where this issue emerged and when, and then what the outcomes were in the COP in 2018 in the COP MOP, and provide an overview and the outcomes of the process of the current biennium 2019-2020. So, as many of you may have been in Cancun, I imagine, hopefully many of you were there in 2016 that are with us today. Digital sequence information emerged initially during discussions on synthetic biology under the convention. And so at the UN Biodiversity Conference in Cancun at COP13, and this was Nagoya's second meeting of parties, it became a cross-cutting issue relevant to discussions under the convention and the Nagoya Protocol. So this resulted in two decisions, one adopted by COP13 and another by COP2, that established a coordinated and non-duplicative process for work on this topic in 2017 and 2018. And I'm going to just show with you what that process consisted of. And basically, the parties decided to consider what any of the potential implications of the use of DSI on genetic resources for the three objectives of the convention and the objective of the Nagoya Protocol. And to do that, a process was established, consisted of the following steps. There was a submission of views on the implications of the use of DSI for the three objectives of the CBD. So parties, indigenous people and local communities and other stakeholders were invited to submit their views. Then there was a fact-finding study and scoping study to clarify terminology and concepts and to assess the extent and the terms and conditions of the use of digital sequence information on genetic resources in the context of the convention and the Nagoya Protocol. And then those views and the fact-finding and scoping study were considered, as David mentioned, by an ad hoc technical expert group, and this took place in February of 2018. And that group was mandated to examine the implications on the three objectives of the CBD, and also to identify different types of digital sequence information on genetic resources that are relevant to the convention and the protocol, and to consider the technical scope and legal and scientific implications of existing terminology related to DSI. So all that work was carried out, and then the AHTEG outcomes were considered by the subsidiary body on scientific, technical, and technological advice at its 24th meeting. And that produced a recommendation that went to COP14 and COP3. So then what happened in Egypt? As many of you know, the conference in 2018 took place in the beautiful city of Sharm el-Sheikh in Egypt. And there we had two decisions, one for the convention and one for the Nagoya Protocol. And in these, the COP and the COP-MOP recognized the importance of DSI on genetic resources for the three objectives of the CBD. Also recognize that access to and use of DSI on genetic resources contributes to scientific research as well as to other non-commercial and commercial activities, and that further capacity to access, use, generate, and analyze DSI is needed in many countries. Since there was a divergence of views among parties regarding benefit sharing from the use of digital sequence information, parties in Sharm el-Sheikh committed to working towards resolving this divergence through a science and policy process that was established for the current biennium. So these are the steps of what we've been doing in the current biennium. Again, there was a call for submission of views. This was a bit broader. It included views to parties, IPLCs, and other stakeholders were invited to provide views to help clarify the concept, terminology, and scope of DSI. Also on domestic measures on access and benefit sharing, on benefit sharing arrangements from commercial and non-commercial use of DSI, and then on capacity building needs regarding access, use, generation, and analysis of DSI. In addition, as was mentioned by David, we had peer-reviewed studies, and in this biennium, we had 4 studies. The first study was on concept, scope, and use of digital sequence information. Then we had 1 study on databases and another on traceability that we combined into 1 study. And we had another study on domestic measures, and it was quite challenging in the time frame that we had to commission these studies and get them done We're very grateful to the team of researchers that's with us today that carried out this work. They had a very narrow time frame to do this. I think we started perhaps in May 2019, and they had to have their first versions of the papers by June or July, and then we worked on them, and there was a peer review. They were finally posted in November, December 2019. And those studies, which you'll hear about in the first block of the webinar, went to the AHTEG. So the AHTEG on DSI, which took place in March of this year, was the first virtual AHTEG. And it was mandated to develop options for operational terms and identify key areas for capacity building. Oh, and also the AHTEG considered the implication of these options for operational terms. So this is where we are now. We're at this these 3 steps that we've completed, and because of the global pandemic, we haven't been able to have the open-ended working group consider the outcomes of the AHTEG. Hopefully this is going to happen in the next year, in the first quarter. And then the outcomes of the open-ended working group will be considered by COP15 and COP4. Just to share with you that we have posted everything online. You're welcome to visit our website to download the peer review studies. You can also see the comments that were submitted during the peer review and how these comments were addressed. The same with the AHTEG report and the meeting documents. They're all posted online, and the final report is available for your perusal. I just want to finalize with a couple of words about the open-ended working group. As you know, the open-ended working group was established to develop the post-2020 biodiversity framework. And it's also mandated to consider the outcomes of the AHTEG and to make recommendations to COP15 on how to address digital sequence information in the context of the post-2020 global biodiversity framework. And so the open-ended working group will be then submitting its outcomes to COP15 and to COP4. So this is it with our process, where we're at, and we're going to be sharing with you the studies and the outcomes of the AHTEG. Thank you so much.
Thank you very much, Christina, for this overview. I think the process was very clearly explained. I really like that slide. It made everything clear. So let's dive right in, because we're already running a few minutes behind. So we're going to go into the peer review studies and have the authors present the key findings from those. So the first speaker is Marcel Jasper. He is a professor of organic chemistry at the University of Aberdeen in Scotland, UK. And his research aims at discovering biologically active compounds from natural sources, both using genetic and chemical methods. He's also the co-founder of Ox Therapeutics, which is a company that uses digital sequence information to design and generate potential drugs. So Marcel, if you can take it away, and you have 10 minutes.
How do I share my screen? I've lost the ability.
I will. You should be able now. How's that? We see your screen now.
Which screen do you see?
A shark.
That's the wrong screen. Okay. Okay, change.
It's a very nice background.
Oh yeah, I know. I'm trying to find a show screen. Okay, that's not helpful at all.
That is still the shark.
Ah.
So when you're in PowerPoint at the... Yeah, there we go.
Okay.
All right, concept, scope, and...
Okay.
Take
I know we tried this yesterday, it still didn't help. Okay. Welcome, everyone. Thank you very much. I'd like to present the work that was done by myself, Dr. Wael Hussein, and Rodrigo Sara on the concept, scope, and current use of digital sequence information. We took as our departure point the RTEC list, but I'll start to introduce what I'm going to talk about today. So first of all, I'll talk about the key issues to be addressed, and I'll try and talk about clarifying the concept and scope, going from the original list and talking about this concept of a flow of data and information from a genetic resource, and then following up then the idea of what the groupings are, and then what those implications could be, and some clarifications at the end of it all. So the key issues, really it's how do we distinguish between different types of data or information. And on the one side, it's the idea of the extent of biological processing that happens when a genetic resource is turned into digital sequence information. And again, the steps that it takes and the proximity to that genetic resource matters in terms of how you look at it. So the question is, how far can be extended? Is it just the DNA information, or also RNA, protein sequences, and metabolites? And what about the additional associated data that goes with that? I would like to then mention the current terminology and how it fits, and that we propose an additional grouping in the middle, because the current terminologies are either very narrow or very broad. What we're introducing here is the concept of those intermediate groupings, which may be helpful to further the discussion. So the original list was very comprehensive. It was prepared by the RTAG that was mentioned by Christina earlier. And to a chemist like myself, who looks at structures on a daily basis, this divides neatly into two different categories. The main category to me is the molecular data. All of the concepts on the RTEG list from A to D involve molecular data. For instance, the nucleic acid sequence reads down to the macromolecules and cellular metabolites. And again, beyond that point, what we have is this information that comes distant, much more remote from the original genetic resource, and is only tangentially related to the digital sequence information. So we'll talk about that in a minute, how those could be dealt with the new groupings that we're proposing. So the normal flow is we take a genetic resource from which we obtain perhaps some metadata that might be the data associated with the sampling site, the organism itself, its taxonomy, etc. From that, we obtain a DNA through a process that was described by my colleague last week. And that leads to DNA sequences and sometimes functional annotations, so understanding of what those genes do for the organism, and that subject is termed genomics. RNA is derived from that, and those sequences give us information on gene expression data, and that subject is typically referred to as transcriptomics. RNA gives rise to proteins and things like enzymes and structural proteins in organisms. And those sequences can be derived either by translation of the RNA sequence information, but also each of these proteins has a three-dimensional structure, which again is additional information that needs to be derived through a great deal of additional experimentation. Finally, we have the metabolites, which are often produced by the proteins. There's different categories, and you can identify those molecules and by just using techniques such as mass spectrometry, or you can derive the small molecule structures using more sophisticated techniques such as crystallography and nuclear magnetic resonance. That subject is often referred to as metabolomics. So the question is, how do we define that? Under the INTSC, the database consortium on nucleotide sequence data, we referred to the DNA and RNA as nucleotide sequence data. You might refer, for instance, to anything that has molecules in a sequence as sequence data. So a protein might also be regarded as sequence data just by the definition of the word sequence. And then under the nucleotide sequence data definition, we also have this understanding of subsidiary information, which is information that is outside of the original nucleotide sequence data, and it could be information on the genetic resource itself or on the proteins and the metabolites that derive from that. So we saw that the proximity to the genetic resource and the extent of processing in the biological system can be used to propose a number of subject matter groupings, and here they are. So we define them in different ways. First of all, you have this genetic resource and associated information. And we go along the line from there, and the further you get to the right, the more remote you get from the genetic resource. And the original grouping that we see is the narrow, very defined grouping containing only the nucleotide sequence data. The intermediate groupings, we have two proposals. The first proposal is that you include DNA, RNA, and proteins. And then the intermediate grouping 2 also includes metabolites. Finally, the broad and inclusive group would contain all the information associated with the genetic resource, so all the molecular data as well as all the other data that the RTAG outlined. But I would make a special case here that we should separate the subsidiary information for the genetic resource, particularly taxonomic data and sample data, sample source data, such as location, GPS location, and conditions, from things like traditional knowledge, ecological interactions, photographs of the organism, and other types of information that were included in the big list by the RTAG. So what we've done as well is we've looked at this in a different way. I decided that a good way to do this would be to put it on two axes. The first axis along the bottom is that extent of proximity to the genetic resource. The further you get to the right, the further you are from the genetic resource. On the other side, other axis, I put extent of processing. This is the amount of additional work that's needed to turn that original data into information. So group 1 would be, for instance, just DNA and RNA information and nothing else. Group 2 and 2A would be, for instance, the sequence data, including protein sequence data, but also information on sequence assembly, structural annotation, and genetic mapping. Okay, so that means that the extended processing is not huge, but it's still substantially different from the original group 1. For group 2B, which is another intermediate grouping, we decided to include functional annotation of genes, gene expression information, epigenetic data, and molecular structure of proteins. Again, molecular structure of proteins is still not a trivial set of data to obtain. It takes either X-ray crystallography or electron microscopy to obtain that information. Again, it requires a great deal of additional processing and experimental verification. Group 3 would be the same as group 2B, but includes data on metabolites, and that would include their molecular structures in this case, so molecular structures of small molecules in biological systems. And finally, the inclusive grouping is group 4, and that contains all the information, including all the elements that the RTAG list refers to. So up to group 3, it's molecular data. Group 4 includes all the other data too. So this is a very busy table, but it's useful in the original report as well. The reason for that is that it actually shows you very clearly that the original narrow and defined groups that were defined by a number of different publications and reports gives you things like digital sequence data, genetic resource sequence data, genetic sequence information, nucleotide sequence data, and we've mapped it out against the RTEC categories, which have been subdivided for clarity. For instance, in some cases, things like functional annotation has been taken out of the original B category. Molecular structures of protein has been extracted from category D for clarification. What you see then as well is that under the broad and inclusive group, it pretty much includes everything, although some definitions are less clear. What you see is our introduction of the intermediate groups really helps to clarify what is included and what is excluded in a very clear sense. And again, the difference between, for instance, groups 2B and 3 is the addition of this molecular data on structural data on proteins and macromolecules. And so it's a big difference to either the very narrow or the very broad. It brings a very nuanced view of how you might look at these different groupings and how they might be useful. So the terminology.
You have 1 minute left, Marcel.
Thank you. Nearly there. So the terminology then used for DSI, if you look at it, the previous information, genetic resource sequence data would have been there. Nucleotide sequence data would have been roughly there. Subsidiary information to nucleotide sequence data is that additional information. And finally, the in silico data that's used by the BBNJ process is the information around the outside of everything. So finally, I'd like to show you where the intermediate groupings fit there. So the intermediate groupings are the molecular data that fits in to that group. So this is the space within which I feel definitions of digital sequence information should reside, rather than the very broad category out here. So this defines it clearly within the limits of this axis here. extent of distance from the nucleotide, distance from the genetic resource, and this size extent of processing. So finally, just to show how I feel that those groupings would apply to the different types of applications. And for group 1, everything is important to most sectors, and we've just chosen 4 representative sectors that use DSI. Finally, here, group 2, again, would be really critical to the last 3 sectors. And then as we go down the list, it becomes a little bit less critical as you go along. So I would like to stop there, and if you have any further questions, they can be posed either in the chat or in the question and answer session later on. Thank you very much.
Thank you so much, Marcel. I think this talk was one that people were awaiting one of the most, with the very clear proposition for defining DSI in a very clear graphical way. I think it's very interesting, very informative. Yeah. And thank you very much for that.
Okay.
We'll move on to the next. Yes,
one clarification, just to say that the RTAG I refer to repeatedly is the original RTAG, not the second RTAG that was meeting in March this year. Just for anybody.
Thank you. Thank you for that clarification. So we'll jump into the next presenter, Amber Hartman-Schultz. So Amber got a PhD in microbial genomics from Johns Hopkins University in the USA. And for the past 10 years, she's been working at the interface between science and policy in California and the US federal government. She's now at the Leibniz Institute, and she is spearheading the international and strategic activities with a focus on access and benefit sharing and DSI issues. And she's including the process to become the first registered collection under the EU implementation of the Nagoya Protocol. Amber, the floor is yours.
All right. Let's see. Did the share screen work? Is it the right one?
Yes, we can see your presentation very well.
Sorry, remember this is just a bit awkward to figure out which is where. Okay.
No worries.
Thank you, Charlotte. Thank you, CBD Secretariat and AHTEG members, for the chance to speak to you this afternoon, this morning, wherever you might be this evening. I'm going to give you a whirlwind tour through sequence databases and the traceability of some of the sequence data that Marcel introduced for you. The first thing I wanted to do is to highlight for you the terms of reference for our study. In the peer review process, we did receive some feedback and some concern that our definition of DSI for the purposes of our study was perhaps too narrow. And so what I've done for you here, in particular in the second paragraph, is to highlight the fact that our terms of reference required us to report quantitative data on the size of the databases, number of accessions, their origin, and the providers and users. And this quantifiable data is actually only possible if we know what kind of data we're talking about. And so as a result, we basically had to make an executive decision in coordination, of course, with the Secretariat to figure out what kinds of data we would be looking at. And it's also really important to note as study authors that we had only 3 months to generate that very first draft back to the Secretariat. So a very short amount of time to do these global quantitative bioinformatics analyses. And so we chose, for the purposes of this study, without bias for the outcome of the AHTEG, to use a known defined scientific term, and that is nucleotide sequence data, which we will abbreviate here as NSD, which is a proxy for DSI, but of course, depending upon how that term is ultimately defined, might be an imperfect match. We also note in the study that the database processes and the infrastructure for sequence data, for nucleotide sequence data, is actually quite similar for protein sequences. And so lessons learned here are probably extendable to protein sequence data as well. So with that introduction, I'd like to tell you where we started our database analysis. The first stop for us was this journal called Nucleic Acids Research. This is a leading journal in the field of databases, and every year, once a year, they produce a database issue where they take an inventory and an update on all biological databases that exist known to this community. And looking at that, we have to realize appreciate how big the biological database space is. Almost 1,800 biological databases is the size of the world of data for biology, and about 800 that focus on nucleotide sequence data. And about 50 of those focus on human data. And so for the purposes of CBD discussions, we don't want to look at those, which leaves us with about 750 databases that are important for non-human nucleotide sequence data. When we looked into these, we manually went through these with a great team of students here at the DSMZ, and we were able to find out that 95% of these databases directly link to or download the data from something called the INTSC. More in the next few slides. That's the International Nucleotide Sequence Database Collaboration. So that shows you how many are sort of depending upon this core infrastructure. We then looked at those that did not directly link to or download from INTSC, and we found there are 38 of them, but actually they all rely upon the unique identifiers, which are called accession numbers, that are generated by the INTSC. And so they are indirectly linked. They do allow upload, but they of sequence data, but they also require accession numbers indicating their indirect linkage to INTSC. We also looked specifically because this group allows upload, so has some degree of independence from the INTSC, at the access policies of these databases, and found that none of them actually had access policies except for one. And this limited the use of the database to non-commercial purposes only. Ultimately, the take-home message from this biological database inventory was to show that 99.9% of public non-human sequence databases rely on the INTSC. And so now, of course, you need to know a bit more about the INTSC. This is a 40-year-old collaboration between 3 scientific institutes, GenBank in the US, the European Nucleotide Archive in Europe, with physical headquarters in the UK, and the DNA database of Japan. You can see these arrows here in the middle indicate something important, which is that every 24 hours, the data sets of these 3 separate databases are exchanged and mirrored with each other, creating this core infrastructure and creating another layer of data security. To create this infrastructure costs about $50 million per year. And this infrastructure is free to all users, does not require registration, and also flows into, as I previously mentioned, about 800 plus databases every day, every night, every month. So what is the access and use policy of this core infrastructure? It's very important and actually has for science at least, a relatively old tradition at this point. That is free and unrestricted access to this data. They declare that they will not attach statements to records that restrict access to the data, that these records will remain permanent and fully disclosed to the public. Now, what's important, if you think about the dependency on the INTSC of these 800 or 1,800 downstream databases, they actually inherit this access policy because this data, the sequence data DSI, was delivered to them from the INTSC, then the databases essentially inherit the access policy of the INTSC. So now perhaps you're wondering why this big infrastructure, why this $50 million annual investment, and why do we do this, this disclosure into these big open databases? Well, there's at least 4 good reasons that we outlined in the study. Number 1, scientists must publish. It's publish or perish. That's how it works. And we are not able to publish our papers unless the data has already been made publicly available in the INTSC. The grants that we receive to do our research often require it. It's part of the grant agreement that we sign in order to get the money to do the research. It's also part of scientific ethics and scientific integrity. It enables scientific reproducibility and also data security. And 4th, and this is an incredibly important reason that's perhaps not immediately intuitive. If you read the letters ACGT, ACGT, this means nothing without context. And we talked about this last week in the first webinar. the databases provide the ability for comparison, and only through comparison can we understand what new sequences mean. So what kinds of stuff is in there? Well, every branch on the tree of life has a representation in these large databases. Of course, the genome sizes of these organisms are all different. And so the proportion based on base pairs can be different according to the organisms. So some genomes are very large, some are smaller. We also note that about 11.5% of the database is human sequence data, so that would be outside the scope of the CBD. There are also model organisms that have been lab strains, lab organisms for multiple decades, if not centuries, synthetic organisms, unidentified organisms, and of course, plants that will have different legal provenances, which means the take-home message, there is stuff in there that's outside of the CBD scope. There is stuff in there that is in the scope of other ABS and other future ABS agreements. So what about the user statistics? More data, and here you're going to get a couple of world maps. There are about 10 to 15 million total users of the INTSC, and they are in every country in the world, including the Vatican, we learned. The heavy lifters here are the United States, China, India, and Japan, and you will see that this reflects also in the next map. If you take into account the $15 million annual price tag, you can break this down per user, which means that it's about $3 to $5 per user per year. And about half of the users live in countries whose governments do not contribute to the infrastructure costs. One might say this is a type of benefit sharing, but the point being that the system is open and free and paid for by a few. Where actually is this sequence data sourced from? Where is the original genetic resource from that enables this open data? Again, here, the heavy lifters are the countries where there's also the most use. So the physical sourcing of this DSI is China, United States, Canada, Japan. In fact, of the data that has a country tag or country information in the database, over half comes from just 4 countries. And this is also very important when considering future policy mechanisms. So you can take these last 2 maps, users and contribution of DSI to the database, and ask what is the relationship between these. And suddenly countries like the US and China become neutral. They become middle of the road. They have similar users and similar contribution of DSI to the database. And now here we see the countries that are more dominantly using than contributing their DSI. So countries like Lebanon, Ukraine, Belarus, Iraq, Colombia, Algeria, that use more than actually contribute DSI to the databases. Another interesting question, and here again, some of the old players come back into the maps, is to ask who is doing bulk downloads of the entire INTSC database. This is possible through a technical data transfer system called FTP, and it's also possible to track where those IP addresses come from. And here you see again, mostly developed economies that are doing these bulk downloads of the entire INTSC data set. We were also asked to look at private databases. Of course, private is inherently private, and so it's very hard to figure out what's in here. And so we reached out to individual companies and asked to do interviews. 6 agreed to do that in the 3-month time frame that we had. And as you can see here, the take-home message from these case study analyses is that all companies download INTSC dataset via the FTP mechanism, which I just mentioned. They all generate sequence data, and they all submit their data to the INTSC. Another interesting aspect, of course, is patent data, data that is mentioned in a patent application. About 20% of sequence entries in the INTSC come from patent or are associated with a patent. And actually, it's a requirement of at least 3 patent offices that the patent applicant upload the sequence data to the INTSC in order for maximum transparency and reproducibility for the patent itself. We also found out that these patent sequences do not have geographical information in them. Another reason why, one of the reasons why the 16% number that I mentioned earlier it happens. So that's the landscape, if you will, of the databases. And now I'd like to sort of turn to the issue of traceability. Of course, this is deeply related to the databases because one important starting point for understanding traceability is to look at how the databases are, of course, connected. So let's start there. Here's a graphic we tried to create to explain what the existing traceability system is around DSI, around sequence data. So the user might start on the left-hand side with a genetic resource. So I picked my favorite organism, an archaea, a single-cell organism called Halophyrex volcanii, obtained from the Mediterranean Sea. And this organism would have been chemically broken apart. The sequence data would have been extracted. The DNA molecule itself would have been extracted and then placed on a sequencing machine. And ultimately, that sequence data would have been uploaded to one of the 3 INTSC databases. And as you can see here at the top, this was a GenBank entry that I pulled for you here. And you see the date of submission along with a lot of other metadata. The sequence data itself looks like this. So here are the letters of DNA, A, C's, G's, and T's. And as you can see up here, this is actually protein sequence data. So this is to reference what I mentioned earlier, that what's true of DNA sequences is often going to be true of protein sequences, although downstream databases will differ. After it's uploaded into the INTSC, the authors will then write their publication, and then when they go to publish it, the INTSC will release that data to the public, and the link will be made to the scientific publication, and that's also possible through the use of a digital object identifier, or a DOI, as you can see here. And then this publication within it will also list the accession numbers that they receive from the INTSC. So here you can see these numbers, CP001953, those are numbers that we received upon submitting our sequence data to the INTSC, and then the journal basically says, we won't publish your data until you provide these unique ideas. And that's those are the simple steps that we have in the scientific community to establish traceability through the database ecosystem. So from now on, these sequences will be known under these accession numbers. And then downstream, here are these 1,800 biological databases that will pull data back and forth to the INTSC. We can ask other questions, for example, around microbial taxonomy. I need to speed up again, which is always a danger. So then what about other traceability lessons from other technologies? Well, we looked into the music and video industry and noted that there is not a good metaphor here because these are rather passive uses of the data. Bioinformaticians, biologists, we interact and touch the data, and we don't just sit there like you do with Spotify or with Netflix and just receive data. We actually need to interact with that data. So those are perhaps not good models, we conclude. The ideas of cloud genomics are promising. However, we note that these are closed systems that require prior knowledge of what kinds of analyses the user will want to do. So this might be very difficult in the CBD community where we have very diverse users. We also looked at blockchain, which has received a lot of attention in the last few years because of its application to cryptocurrencies like Bitcoin. And we asked the question here, what are the requirements for a blockchain system and what are some perhaps challenges when applying it to the CBD community? Blockchain is not an inexpensive technology to build, in particular, not in terms of its energy costs, as well as technology costs. And so the CBD would need to address funding costs and whether or not there are enough users that would use the system that would make it worth it. We note that it's probably not compatible with the INTSC, at least in its present form, which might mean you might reinvent this wheel. As I explained, it's a very complex and integrated system that already exists. And we also note that scientists would likely not be able to publish on data in a closed system, given current requirements. We also asked the question, would the sequence data itself stay in the blockchain? And if so, you would need to know the analyses that would have to be done in the blockchain. And this is very challenging to know a priori. There are also significant size limits to blockchain. And with human data sets, it's actually possible because 99.9% of the human genome is the same between humans, to use blockchains for human genetic data, for patient data. But that's only possible because we throw away all of the repetitive data. For CBD-related data, you can't throw away the data because it's exactly that diversity that you would want to capture, and so that creates a very real challenge. Perhaps most importantly was one of the experts that we spoke to noted that the user needs to be motivated to use the blockchain system. So if we look at Bitcoin, the cryptocurrency based on blockchain, if you take a Bitcoin outside of the blockchain, it has no value. It doesn't have any currency value anymore. However, if you remove sequence data from the blockchain, which would be very easily possible, the value is not lost. It's still sequence data. And therefore, how you motivate the user to stay inside the blockchain could be quite challenging. Sorry, Charlotte, 2 more slides. Technical observations, just to quickly summarize, 16% of entries reported country information in the databases. 44% of entries did not report a country, but should have. 4% of entries are linked to genetic resources, mostly held by public collections. Digital objects, for example, like internationally recognized certificates of compliance, could theoretically be linked to sequence data, but unpublished private documents like PICs and MATs, not in the form of IRCCs, are not amenable to connecting to sequence data. Information in patent applications is not fully transferred to INTSC. In particular, country information is not transferred. The lessons learned here, the take-home messages, the majority of sequence data is out of CBD's scope, as I explained, for both material, geographic, and temporal information, scope reasons. The core infrastructure for sequence data is the INTSC. The ecosystem is big and complex. Traceability within databases is possible, is already being done, but after it's removed from the system, the traceability system breaks down. And the system, INTSC from sequence data from INTSC is used in a patent application. This is also not traceable. And finally, on blockchain, the size of the current global data set, which is exponentially growing, is not manageable with current technologies from blockchain. This is the team that put this together, and thank you for your patience.
Thank you very much, Amber. And to be fair to you, you had 2 studies to combine into one talk. So that was a lot of information. Just to remind everybody, so these studies are posted on our website, and maybe somebody can, we'll put them in the chat. We'll put the links to the studies so you can all go back and look at this deluge of information and look at it, you know, in your own time a little bit later. We're going to move on because we're running a little behind. So the next speaker and the last one for the studies is Margot Bagley. So Margot is an intellectual property law expert, and she's also a professor and chair at the Emory University School of Law. And she has authored and co-authored several studies and reports on digital sequence information for the Secretariat for the Convention on Biological Diversity, the Plant Treaty for the FAO, and other organizations concerned with DSI. Margot, the floor is yours.
Thank you, Charlotte. I appreciate the introduction. It's my pleasure to present the results of the DSI study number 4, which is a fact-finding study on how domestic measures address benefit sharing arising from commercial and non-commercial use of digital sequence information on genetic resources, and address the use of digital sequence information on genetic resources for research and development. We had a long title for our study. I want to start by noting that the study was compiled by a wonderful team of main and contributing authors who worked very hard in a very compressed data gathering time period to identify countries with domestic measures relating to DSI and various approaches that are being taken to DSI and ABS. And several of the contributing authors also drafted case studies that are included in the annex of the study that provide illustrative approaches to DSI utilization. A brief word on methodology. So we sought information from all 196 parties to the CBD through a combination of a survey that the Secretariat sent for us to all of the national focal points, a review of submissions that were submitted in the process from parties and other interested entities, interviews that team members conducted, analysis of information available in the ABS clearinghouse, a literature search. We were just, and other websites as well, we were scouring, trying to find information on these domestic measures. And in terms of what I mean by domestic measures, formal ABS legislative, administrative, or policy measures, such as laws, regulations, decrees, proclamations, ordinances, policy statements, codes of conduct, guidelines, best practices. We took a very broad view of what could be a DSI domestic measure in terms of compiling the results of this study. And I will also note that we did have some challenges in collecting the data. DSI is a politically sensitive topic, and even within some countries, there's not a consensus on what the proper approach should be. There were issues of confidentiality when speaking with folks. Some of the information was incomplete in the clearinghouse, and even in some of the survey results received and sometimes conflicting information. So we had to make some interpretive assumptions in some cases. Nevertheless, we persisted. So key findings from the study, we found that 16 countries and 1 subnational jurisdiction have domestic measures in place that address DSI. 1 country addresses DSI by ABS implementing measures in the absence of formal domestic measures. And 18 countries that at the time of the study did not have domestic measures indicated that they are in the process of developing or have plans to introduce such measures in the future. We note that none of the parties indicated that they had received monetary benefits from DSI to date. In terms of terminology, what were we looking for in terms of DSI? We found that countries or parties generally took one of 2 approaches. They either used specific terminology that covers DSI type subject matter, such as genetic information, genetic heritage, intangible components, or they interpreted existing terminology to include DSI, such as genetic resources or genetic material. And so those were the kind of terminology approaches we saw. We were able to group countries with domestic measures into 5 basic categories, though I will say that some countries didn't fit neatly into any single group. The 5 basic approaches are 1, DSI is only addressed in conjunction with the utilization of physical, tangible genetic resources. 2, PIC and MAT are required for DSI independent of access to a physical genetic resource. 3, benefit sharing obligations arise from the use of DSI, but there are no access requirements for DSI. So it's just utilization that triggers benefit sharing. 4, some parties address DSI even indirectly through compliance or monitoring mechanisms. I'll say more about that in a moment. And then we found intentional non-measures to promote unrestricted access to and use of DSI. So instead of domestic measures on DSI, measures basically encouraging the use of DSI without restriction. So to briefly go through these approaches in just a bit more detail, approach number 1, where DSI is only addressed in conjunction with the utilization of physical genetic resources. There, a country would have access provisions in their ABS framework for genetic resources, not separate access provisions for DSI, but through permits, contracts, mutually agreed terms, restrictions might be placed on the use and further distribution of DSI. The second approach, for these countries, physical or access to a physical genetic resource is not required for there to be access requirements on DSI. So access, or the term genetic resources, is interpreted broadly enough to cover DSI which in turn potentially triggers pick and map obligations. And I will note that even though countries have these measures, it does not mean that they are all in operation at the current time and that they are actively requiring access. But we did see evidence of these domestic measures. Approach 3 does not require access for DSI, access provisions for DSI or for physical material, but benefit sharing is triggered by certain actions. And DSI in databases such as Amber just talked about is considered to be within the scope of these measures. So open access, DSI not restricted, but benefit sharing required. In terms of compliance and monitoring, some parties that do not otherwise have measures that address DSI have compliance measures that indicate that users should respect conditions in mutually agreed terms and material transfer agreements, including those that deal with DSI. So this is an indirect compliance obligation in relation to DSI. In terms of monitoring, we have, for example, one country, India, that monitors patent applications worldwide and has benefit sharing requirements for DSI from Indian species that show up in patent applications. So that's a monitoring type of approach. What does it mean when there are no domestic measures on DSI? Well, of course, the vast majority of countries do not have domestic measures on DSI. And two primary scenarios. One, and I guess the first one could be three, really. No measures yet, but either there's an intention to adopt measures in the future, or countries are not sure and kind of waiting to see what happens at the international level in the COP-MOP before deciding whether or not to move forward with DSI domestic measures. And then there are countries that have, as I mentioned, intentional non-measures, no intention to regulate DSI access or benefit sharing. And these non-measures promote public access to DSI in open access databases. It's a deliberate policy decision to have no regulations on DSI access or benefit sharing. ABS implementing measures are often the tools that are used to operationalize the domestic measures relating to DSI. So contracts and permits will have terms that relate to DSI, how it's used, how it can be used, how it can be further distributed, et cetera. Benefit sharing, as I mentioned, no direct monetary benefit sharing reported so far. Non-monetary benefit sharing generally relates to the kind of generalized benefits from the fact that DSI is openly accessible. And then in one of the case studies, it was noted that there are a variety of non-monetary benefits that are expected to result from DSI, such as open access publications, capacity building among African scientists in that particular case. So non-monetary benefits, yes, and monetary benefits expected, but not yet received. So implications of the differences in approaches, well, in some cases you have access limitations on DSI, and others only benefit sharing obligations. And those differences have implications for transaction costs under the bilateral model of negotiation, for traceability, when you think of the numbers of databases and the inability to trace third-party uses, and for open access to DSI if access limitations are put in place. Finally, the study concludes with a number of annexes. One has a table of jurisdictions with the specific measures on DSI and benefit sharing listed. There are 5 case studies exemplifying ways that DSI and benefit sharing are being addressed. They're the results of a literature search, the survey instrument, just a variety of additional information in the annexes. So to conclude, there are a number of parties that are currently employing domestic measures in relation to DSI and benefit sharing, and the possibility of other countries doing so as well. And I will end there.
Thank you very much, Margot. This was a very good overview, and I really thank you for this presentation. Very informative, very interesting. I think we actually have some questions. So we are running a little bit behind, and I think the most important thing is for everybody to be able to present. So we're gonna have just 10 minutes for the Q&A at this point, and I will ask our panelists to turn on their webcams and turn on their microphones at this point when they're speaking, and to keep your answers short and concise, 1 minute, maximum 2, so that each of you can actually have one question. And I will give the floor to my colleague, Christina, who has been filtering through the questions. Thank you.
Okay, thank you, everybody. These were excellent presentations. We've been receiving several questions that then we're assigning to the different panelists to answer if it's something quick and easy. But we also have some other questions that we would like to pose to you, maybe one per person because we're running a little bit late. So I'm going to start off with Marcel. Marcel, how do you see the groupings being used, the groupings that you presented being used to develop sensible boundaries for what could be considered to fall under DSI? If you could elaborate a little bit on that. Thank you.
Is it okay to bring up a slide for that picture?
I think so, yeah.
Just need to, here we go, show my screen again. Which screen can you see again?
It's good.
The clarification slide.
Okay,
Yeah.
good.
Mm-hmm.
So there was two answers that I have for that. Really, is the first one is this this one here, and it's talking about how you might incorporate changes in science over time, and this is something that's happened fairly recently. So that we see these things advancing, and the way that you see the groupings then is that under normal circumstances we would have this idea of having the ability to process data automatically to get, for instance, DNA annotations, many of which need to be verified by laboratory work. But again, RNA sequences can be automatically annotated, and from that you can derive protein sequences automatically. However, something like a 3D protein structure is, until now, very difficult to obtain without significant experimental work. So what's happened in the last week or so is that this publication came out that showed we can now predict with 90% certainty the structure of a protein. And that's really a massive advance. This is an AI system that relies on previous structures and a sort of expert system. That means that we can actually change the label here for protein structures from red to green, or partially automated to an orange label. And that allows me to think in a little bit different way how we might address the issue of scientific advances. So those groupings at the moment are pretty fixed along the x-axis. So you can look at how far you want to go, and that's a decision to be made by policymakers to see how far you wish to go along DNA, RNA, proteins, or metabolites, and what you wish to include. But on the y-axis, you need to be flexible in the language that you use to describe it, because over time, those boundaries will stretch, and they'll try to make it sort of graphical. What you see then is those groups will expand in the y dimension, but not in the x dimension. We don't expect new life forms to arise that have different non-DNA encoding of their genetics. But on the y-axis, we might well expect that some of the things that we now find very hard and require a lot of experimental work might become routine or automated, such as we saw for the protein structure. So you might well see that the extent of processing changes, and that might be something that a scientific and technical body might want to address every few years as you go along this process. Again, so it might not be something you want to fix at the beginning. You'll say this is the current state of play, and we expect it to change by this much over the next few years. And then finally, you might want to say, well, actually, and in 5 years' time, it's time for a review to see how far science has come during that period of time. So that would be my view of how that might be done. So you keep the intermediate grouping as your main grouping, molecular data, and then you expand that in the y dimension as time goes on and science advances. Thank you.
Thanks so much, Marcel. I'm going to pass it now over to Margot. Margot, we have a question for you. You mentioned Brazil recently changed their approach on digital sequence information in relation to access and benefit sharing. Could you elaborate briefly more about what these changes are? Thank you.
Sure, if you can allow me to share my screen. So Brazil was one of the countries under approach number 3. So, all right. And it was very interesting. We received a detailed explanation of the Brazilian approach from the national focal point that was very helpful for our study. So Brazil has a new registration system in place since 2017 to regulate benefit sharing for genetic resources and DSI, but not access. And the idea is to greatly reduce transaction costs, no need for bilateral case-by-case negotiation. And it includes DSI as genetic heritage and information of genetic origin, including in databases. Registration is online, it's mandatory, but only prior to certain triggering activities, and you don't have to have access permits prior to research activities. Notification and benefit sharing is required before economic exploitation of finished products, and users can choose either monetary benefits, 1% of the annual net revenue paid into a national benefit sharing fund, or non-monetary benefit sharing, and there are a list of activities that are provided that can comprise non-monetary benefit sharing. So in a sense, that's very straightforward for, or at least it's designed to be straightforward for the user. And they have had a number of registrations so far, 47,000 access activities registered, the idea being that if they've made it easier, that they'll have increasing compliance with this. 449 in silico, which could relate to DSI, 67 declared with commercial intention, but no finished DSI products notified as of the time of the study. So there had not been benefit sharing arrangements or monetary benefits as of that time in relation to DSI. So we thought that was an interesting approach in relation to DSI that did deal with DSI independent of physical genetic resources, but did not limit access.
Margot, for that explanation. And we also have a question for Amber now. I'm just gonna focus here a little bit, trying to go through the questions. So Amber, your maps always generate a lot of interest when you present the study. So maybe, and there was a couple of questions also in the chat about, you know, the tag or the origin where the DSI is sourced, the country of origin, etc. But maybe you can explain that a little bit. You know, you're saying that over half of the DSI comes from poor countries, but again, only 16% of the sequence entries had an actual country of origin where it was collected. So maybe you can elaborate a little bit so that people can better understand how this was done and how the team was able to figure out, extrapolate to the rest of the data, to the data set. Thank you.
Yeah, do you want me to try to share a screen, or I can,
if you,
is that worth
sure,
it?
if you want to share, if there's something to share, go ahead.
Yeah, it might be helpful. I don't know. I'm going to geek you out here, so bear with me. So this is all these are technical.
And you have 2 minutes, Amber. Just wanted to remind you.
Yeah, you heard geek and you got scared, didn't you? So these are technical details, but they're really important, I do think. So first of all, keep in mind, if 100% is everything in the database, we said about 12% is going to be humans, right? And then 20% is patent data where there is no country information. And then there's going to be model organisms where that mouse was in the lab since 1885 and has been inbred in colonies since then. All that data and things like it are never going to have country information. So we will never expect 100% of the data in the database to have country information, maybe max like a half of it, right? So even then, though, 16% of the total data set is too low. And so what you're seeing here on the screen is we did some manual checks. Like I said, we have great student helpers, and we checked and asked the question first, is that really correct? So we tried, we had 150 sequences, looked at the publications, saw if we had access to the publication, and if we did, we asked the question, is what the author described in the publication coherent with the country information they reported? And in 100% of the cases, the country tag seemed by our interpretations to be correct, meaning we don't think that the maps are wrong based upon wrong country being reported relative to what's described in the publication. Number two, if the country information was missing, should it have been reported? And we basically found yes, in 44% of the cases, it should have been. And in the other cases, there were good biological reasons, we thought, of why the country information wouldn't have made sense to be reported. Then the third question that we asked, because you think, okay, why are these scientists not reporting this country data? What do they have to hide? Well, as scientists, we also know, like in any field, there are lazy scientists and there are good scientists, or we all have a little bit of both. And we asked the question, where the country data was missing, what were the geographical distribution patterns of that missing data? And we basically saw a similar geographical distribution as we saw in the original maps, meaning that it looks like the scientists were careless and did not fill in the country information, but not as though they were deliberately being deceptive, right? So if they were trying to avoid, well, then all of those reporting on US country data where there are definitely no access and benefit sharing restrictions, there would be no reason to hide the fact that it came from the US. They just didn't report it. So these are approximations based upon the best available data that we have. The database is messy. It's a scientific tool. It's not going to be 100% perfect, but we're doing the best we can to give you a sense of where this is coming from. The chat also noted, this is interesting, Helena Paul says, because these are not necessarily obvious centers of biodiversity. But yet this actually doesn't surprise me personally at all. Why? Because there's a lot of biodiversity out there, and it is very expensive actually to go abroad into the deep jungle to do sampling campaigns. We need to do more of this, but yet that a lot of the biodiversity in the world has simply still not been discovered or sequenced or accessed.
Thank you so much.
Thank you very much, Amber. Yeah, and
Back to you, Charlotte.
Thank you very much. Thank you for 3 minutes of geeking out. I could do an hour, but we don't have time there. I really want to respect that everybody should be able to present before 11. So we're gonna stop this question and answer session now. And you still have a chance to post questions in the questions box, and panelists will answer them as appropriate until the end of the webinar. So thank you very much to our panelists. This was great, and we will move on to the second part of the webinar, which is the key outcomes from the AHTEG report, the AHTEG group work and their report. So we're very lucky today that we have both co-chairs who are here to present their findings. So the first person who will the first co-chair who will take the floor is Chris Lyell. He's a scientific associate at the Natural History Museum in London. Welcome, Chris. He is a taxonomist, and he's the UK Global Taxonomy Initiative focal point as well. He has many years of experience working with ABS issues institutionally, nationally, and internationally. He was the chair of the ABS Clearinghouse from its inception in 2018, and more recently the co-chair of the AHTEG group on DSI, and this is the function you're here for today. Chris, the floor is yours.
Thank you very much, Charlotte. Can I just confirm you can see the presentation?
Yes, we can.
Good. So what I'm going to talk about, and Letitia after me, is what the RTEC came up with. So broadly, we're going to talk about, initially, just describe who was part of the RTEC and how that worked, what we were supposed to be doing, and then some key outcomes. Now, what's going to happen in the next 20 minutes or slightly less perhaps, is we're going to try and condense 4 days of intensive work and a quite dense document into a very few slides. This was, as has been said earlier, a virtual meeting. We had it just as COVID was beginning to strike in early March. And at the last minute, the Secretariat turned it from a physical meeting to a virtual meeting, which had one quite good effect, and that it meant that pretty well every member who'd been nominated to the RTAG and accept it was able to participate. Sometimes there are visa problems at the last minute which prohibit membership, actual participation. This time, because we were doing it virtually, although it was challenging and there were time constraints, we did have pretty well everybody there. And as you see from that, there is a range of participation from parties, and that's selected by nomination, and it's balanced by region and by gender, from IPLCs and from other organizations, NGO, public, private. So a very good selection of expertise. The RTEC is not a negotiating body. It's a body of experts which have been selected because they know what they're talking about, to come together to really move from the amazing reports that you've just heard into documents which are perhaps more accessible for the negotiators to deal with, and along the way make recommendations based on those documents. And also, of course, we had the submissions from parties and from other organizations which had been provided to the Secretariat. So what we were doing was looking at the compilation, the synthesis of views produced by the Secretariat of everything that had come in, looking at those peer-reviewed studies which you've heard about in the last session. And then the first thing was develop options for the operational terms and their implications. One of the problems, as I guess you all know, is that DSI is a placeholder term. And people have been using DSI without necessarily every time being very clear in discussion, in writing, in documentation, precisely what they're talking about. And the different concepts of DSI might have very different implications. And after that, we were looking at the capacity building issues. So here we are, and here's a document, a diagram you've already seen. I make no apology for using one of Marcel's diagrams because they're extraordinarily useful. As I said, there are different concepts, but Marcel's report, Marcel and his team's report, provided an organizational methodology. So we could look at those concepts in some sort of logical way. And as Marcel said, we have got the very simple, if you like, sequence data from DNA and from RNA, protein data a little further away from that initial genetic resource, DNA, and then on to metabolites, and of course all the subsidiary information. But there was a context there which we could use, and with a lot of discussion, and there was a great deal of discussion in the RTAG, we came to this this table of groupings, and as you see it, it is very much what you have already seen in the report on scope. Well, here we have 3 groups, not 4. The groups are nested. So group 1 is basically RNA, DNA sequences. Group 2 includes group 1. Again, think back to the images I've already seen in Marcel's report, but adds on protein data, adds on information about gene expression, just what these sequences, what their properties are in terms of what you see in the organism. It also includes epigenetic modifications, some heritable factors which are outside the nucleus. Things like methylation patterns have been identified in some of the document submissions from parties. And then group 3, which includes both groups 1 and 2, and there we're looking into the metabolites, the other macromolecules found in the cells. So just as you have already seen, these groupings extend further and further away from the simple DNA sequence data. Now you'll notice we have associated information as a separate item there, not put in one of those groups. That was quite deliberate. The RTAG considered that those first 3 groups, those molecular groups, if you like, could be considered digital sequence information. It's not up to the RTAG to say, oh, well, it's just group 1 or it's just group 2. This is information that's provided to negotiators to deal with. But the RTAG felt that the associated information, which includes associated traditional knowledge, might not be considered as DSI. It isn't molecular, it's something else. And although it's valuable, and we're not saying it should be ignored, but it is not molecular, and that was the rationale. When it came to associated traditional knowledge, it was stated by a number of members of the RTAG very strongly and quite correctly that there are very clear obligations to share benefits from the use of associated traditional knowledge. But of course, AHTEG is being discussed separately, but within the CBD framework. So it's not a matter of excluding associated traditional knowledge. It's a matter of putting it where it's being debated by experts in AHTEG. And as I say, this is now down to the negotiators. We were not able to pick easy to use terms for each of those groups. We thought actually at the end of the day, rather than spend ages debating, oh, well, we want this term and that term, we'd use the groupings. But a number of terms are available for each of those groupings. And we have in this table, the RTAG listed some of those terms. And you can, if you look at the report, the peer-reviewed study, the Marcel's team developed, you can see a greater discussion on what each of these terms has been thought to mean by various actors. And I would just reiterate that the RTAG report and all of these peer-reviewed studies are on the CBD website for you to see. I'm going to hand over to my colleague Leticia now to continue this presentation.
Thank you very much, Chris. And I will jump in and take this opportunity to actually introduce Leticia, because I haven't done it so far. So Leticia Chitwamulumoni, I apologize for butchering your last name, she is the Deputy Director at the National Department of Environment, Forestry and Fisheries in South Africa, and a national focal point for the Nagoya Protocol on ABS, access and benefit sharing. She has 15 years of experience working in the field of ABS, is currently the co-chair of the AHTEG group on DSI on genetic resources, and is also a member of the informal advisory committee on the ABS clearinghouse. Leticia, thank you so much for being here today. The floor is yours.
Thank you very much, Charlotte, and thanks for the opportunity to share the outcomes of the AHTEG, and thanks to Chris for starting off with the first part of the outcomes of the AHTEG, which was largely focusing on the options that were developed by the AHTEG in order to clarify the concept and the scope of the operational terms. So now there are other 2 important outcomes coming out of the AHTEG, which is now that we have reduced the groupings into 3. So our mandate was also to now look at the potential implications of the different groupings, focusing on the key elements, and also to identify the key areas of capacity building. And I think you've heard from the previous speakers that issues of capacity building was one of the decision that was adopted, which required the RTEC's attention. So in terms of the potential implication arising from the different groups, the focus was on the traceability aspect of the different types of information, and also on the issues of the INSD on open exchange and the use of DSI, and also concerning the use of DSI and technologies enabled by DSI in life sciences, research and innovation processes, and also the measures governing access, benefit sharing, and compliance. So we were looking at those three groupings and try and have a discussion and come up with some recommendations on their implication, especially looking at these four important aspects that are relevant to DSI. However, overall, the discussion were preliminary in nature, and the reason for that was that most of the implications that were needed for all the groupings was depending on the benefit sharing approach that could be considered to address DSI, which I know that we have not gotten to that stage of deciding on the benefit sharing approach, but I think the RTAG was able to have productive or constructive discussion around the implications and also to indicate that some potential implications, were not discussed in depth and could benefit from further information and consideration. And you can see that already from the RTEC, further work or consideration of some elements on the implication were also identified. And how is that going to be facilitated? I think that is the responsibility of the negotiators to discuss and agree or decide on how they would wish to gather further information and allow in-depth discussion on some of the implications, focusing on these four elements. If we can move to the next slide. So the first potential implication was focusing on the important element of traceability. And the RTAG generally reiterated the value of open access, where some experts noted that open access does not necessarily mean free and unrestricted access. Because some of these publicly accessible databases are functioning using differing terms and conditions of use. And I think from the previous speaker, we were given some ideas of how those open access are managed by other databases. And still under traceability, another important aspect that was discussed was on the ways to improve traceability. And there was a number of suggestions which actually focus on the infrastructure development in the form of creating new field entries that could trigger requirement for some important data or information or records that could assist in terms of traceability. And that was looking at the important aspect that I think Amber touched on, the issues of provider country tags that could be missing in other records. And maybe that is probably because it's not a mandatory field to provide such information in other data set, but it's something that came strongly from the RTEC that it needs to be also given attention in terms of improving the traceability aspect, and also looking at some possibility of linking those systems with some of the existing infrastructures like the ABS clearinghouse where we have the internationally recognized certificates of compliance. However, trying to bring it to the three groupings, the RTAG find it more easier to conduct traceability if DSI is only seen within the first group. And they felt that if you move towards group 2 and 3, then it becomes more difficult to ensure traceability on those DSI. However, they also look at the relevance of traceability, thinking beyond the clarification of the scope and the terminology to say, if we are now bringing the possible benefit sharing agreement approach that could be considered, which system will benefit out of traceability, and you know, like if it's a bilateral system or a multilateral system. So there was a lot of discussion around that to also flag out that traceability may not be relevant in either system, depending on what is going to be decided as a mechanism or a benefit sharing approach. And again, it was also noted that it's important to also receive information from this existing database like the INTSC, so that we have an idea of the kind of information that they receive, and that is important, and even the gaps that exist. I think Amber did touch on some of those aspects. So, but we felt that it was important to have a representative from these databases just to share some of this information that could be useful in terms of traceability. Can we move to the next slide? So now coming to the second category, that is concerning the use of digital sequence information, or DSI, in life sciences research and innovation processes. And you can see there, there's not much that has been captured as part of the outcome, because I think the RTEC agreed that this further discussion is needed regarding this part on the potential implications of the different groupings for the technologies and the sectors in the life sciences. And that was largely because you might have seen from the presentation from Marcel that he showed a slide of the different sectors and how the various grouping are relevant to all of them. But from the RTEC's perspective, the general feeling was that perhaps that component, it should look at the different technologies as opposed to the sectors, because if the technology that is needed for all the three groupings is relevant to the life science research and innovation, and I think that could easily make it easier for the negotiators to have a clear decision on that aspect. So we have actually noted the grouping and the information that was given in terms of the relevant sector as provided, but we need more discussion and you know, on the different technologies that could apply to all of them. And in terms of the ABS compliance, again, the same situation happened, because when you talk of ABS and compliance without having decided on the benefit sharing approach that you will want to focus on, then it was difficult again for the RTEC to come up with specific recommendation that talks to issues of measures governing access, benefit sharing, and compliance. And they felt that more discussion is required in that context. Can we then move on to the next slide? So the next component of the RTAG report is on the identification of key capacity building needs. And the discussion from the RTAG was focusing on capacity building as a form of non-monetary benefits. So in that discussion, generally the RTAG acknowledged that capacity building is critical for DSI, and then capacity of countries to develop indigenous research and identify, understand, and monitor and manage their own biodiversity is very, very much crucial. And they also discuss generally that the capacity building needs under DSI could be integrated in broader capacity building initiatives and strategies under the CBD and the Nagoya Protocol. And there was more emphasis that was put during the discussion on the need for appropriate funding and support for development and maintenance of the scientific infrastructure. And I think Amber did share with us some of the costs that are associated with the maintenance and the creation of the infrastructure. So in terms of the key areas for capacity building needs that were identified by the RTEC, it included general understanding of issues relating to DSI, including the economics of information, understanding the R&D steps along value chains based on genetic resources and DSI, analyzing and processing of big data related to DSI, access and use of international databases, taxonomy, molecular biology application for the DNA, RNA extractions, including processing and uploading to databases, bioinformatics, and data management. So these are the 5 key areas that have been recommended by the RTEC for capacity building needs. But the work did not stop here. We also look at the key stakeholders. If we can move to the next slide. Who are the key important stakeholders for this kind of capacity building? Because we need to have that clarity so that we get the right stakeholders for this kind of capacity building. So we need to build capacity of the national focal points and the regulators, as well as the IPLC, to understand issues relating to DSI. And in doing so, at the national level, we must also ensure engagement and collaboration among different government agencies, because this DSI may not be managed by a national focal point, but there may be other national agencies that are very relevant and crucial for this kind of capacity building. Also, we need universities, ex-situ collections, research institutions, private sectors, and all institutions working on bioinformatics to play a role in terms of capacity building related to DSI. And the RTAG also look at the mode of delivering this kind of capacity needs. If we can move to the next slide. So with those key areas that have been identified and their associated stakeholders, the RTAG felt that on-site and virtual courses and workshops could be utilized as a mode of delivering such capacity building, case studies, exchange of information and experiences, sharing of lessons learned, joint scientific research, technology transfer, scientific visits, partnership and collaborations, including through regional networks, and also support for development of scientific infrastructure, including through regional approaches, and examples were given, such as the CGIR centers and the intercultural dialogue through face-to-face meeting for indigenous peoples and local communities, integration in academic curricula, and also integration in regional and international development agenda. And I think this basically concludes the overall outcomes of RTEC. Thank you.
Thank you so much, Leticia, and Chris, on this second part of the webinar. I think this is the important stuff, you know, the party's opinion and on the work of the AHTEG group and what's been done, what's left to be done, etc. We have several questions that I think are particularly pertinent for you too. If you can just please keep your answers to a couple of minutes each, that would be really helpful. And I will let Christina ask the questions. Thank you. Okay. So Christina is telling me to go ahead and ask this one question she pointed out to me. So it's talking about traditional knowledge and why is traditional knowledge and associated information that might be other than traditional knowledge also not considered as digital sequence information?
Right, as both Marcel and I said, the RTAG considered the associated information, although it's important for interpreting the DSI, the molecular data and information, is not itself part of that. The less you have boundaries around what you're discussing, the more difficult it is to actually discuss it. In this case, the RTAG felt the associated information, because it wasn't molecular on basis, because it was only describing it, was something other than DSI. And the process would better be served if it was clear that what was being discussed was that molecular data. Regarding associated traditional knowledge, yeah, clearly extraordinarily important and a very challenging area. And because it is so important and so challenging, it would seem to be best dealt with separately from DSI. Now, nobody, I think, was saying that, oh, because it can be held by digital media, therefore we didn't worry about it anymore. Quite the reverse. But we felt that it was best dealt separately from the molecular content of DSI. But clearly, associated traditional knowledge is being dealt with and will be dealt with. And it's down to the negotiators again to consider the media by which ATK might be dispersed, but in the context of other discussions on ATK. So we weren't throwing it out of the window. We very much said this is important, but it is something different.
Thank you very much, Chris, for this clarification. I believe we have a question from one of our participants, Alexandra. I believe you have the question because I don't have it under right here.
Yes, I was DSI from public research usually must be published in INTSC databases. On the other side, such DSI may fall under restrictions from PIC, MAT, or similar obligations, and therefore must not be shared freely. Do you think it is possible to meet both requirements?
If I may answer that, and I'm going to take off my RTEC co-chair hat and I'm going to put on my researcher hat. As a researcher, if I access genetic resources, I am bound to abide by the conditions under which I access them. And if that means the data cannot be published, then the data cannot be published. It may also mean that as a researcher, I choose not to make use of that material. It's part of the discussions that somebody accessing material would have with the appropriate authorities in the provider country. And it's part of an ongoing, if you like, scientific discussion. But the RTEC itself did not discuss that. So what you just heard was my personal opinion, not an RTEC outcome.
Thank you very much, Chris, for this answer. Glad you have several hats you can put on, so you can take any question. And I believe we have a last question that might be addressed by Leticia. Maybe you guys can decide. So, in the case of capacity building needs under DSI, in the case it's incorporated into the broader capacity building initiatives or strategies, how do we ensure its effectiveness, apologies, to achieve its goal as non-monetary benefit?
Okay, thank you very much, Charlotte. There are merits in having a coordinated approach to capacity building initiatives under the CBD and the Nagoya Protocol. However, the design of such capacity building activities should allow dedication of specific resources to DSI, because generalization could be contrary to the DSI's capacity building objectives. It should take into account the socio-economic context of provider countries and be designed to contribute to enhancing the endogenous capacities of these countries. And in other words, it should be tailored to meet the needs and aspirations of each country and research institutions. So in my conclusion, going back to what the RTAG was required to do and what parties were requested to provide as part of their contribution to capacity building needs, I would say that access, use, generation and analysis of DSI are key elements of capacity building needs, which if delivered effectively could contribute to the conservation and sustainable use of biodiversity as a form of non-monetary benefits. Thank you.
Thank you very much, Leticia, for this answer. And thank you to both our co-chairs for your participation in answering the questions. It's very much appreciated. I think there were a lot of ideas that were addressed, and some had answers and many didn't, and which is exactly what we need to actually as a transition into this further discussions and what's left to be done, by all parties. So thank you very much. We will now introduce the closing remarks speaker. I just want to have a very quick something to say about the survey that will pop up at the end. So when you log off the webinar, your browser will open with questions, and it would be really helpful for us if you could take a couple of minutes to answer them. You can answer them straight away. You can answer them, leave your browser open and answer them later in the day or tomorrow. And just please take the time to do that. So without further ado, our last person to talk is the head of the ABS unit at the Secretariat for the Convention on Biological Diversity, Takonjo Shem Shikongo, will give the closing remarks for this webinar. Thank you very much. Tauko, the floor is yours. Do we have Tauko here? Okay, it seems that maybe Tauko has some issues with his connection. Tauko, if you're show up, just jump right in. Basically, the closing remarks. Oh, there's Talco to save the day. Thank you. The floor is yours, Talco.
On the outcomes of the RTEC. A week ago, we started this journey of bringing all of us on the same page regarding the very complex and challenging issue of DSI. I would like to thank all the participants, the panelists, the co-chairs of the RTEC, and the Secretariat staff, including all those who assist us to ensure that this seminar took place today. The road ahead of us is long and a potential for many difficulties, but depending on the effort we do, we can make it shorter and easier for ourselves. Our next installment of the DSI webinar series will be scheduled sometimes in 2021 during the first quarter. We have now looked at the basics of DSI, and we've also looked at the technical issues around DSI. You are all familiar with the useful work of the RTEC and are up to date on where the process is under the CBD. And next, we'll proceed to look at some of the policy options and scenarios that are emerging regarding DSI. We look forward to your participation in that webinar too, sometimes in the first quarter of next year. Thank you once more to all of you, and may we have a very blessed day and the rest of the week further. Over to you, Charlotte.
Thank you very much, Shalko, for those concluding remarks. And we all hope to see you for the third webinar. So keep an eye out for this. And the recording for this webinar will be posted on our website in the next few days. I wish you all a great day, great afternoon, great night, and see you soon. Goodbye.